Good morning, good morning. So my name is Rahul Roy. Yes, there's a famous film actor. Do you know about him? No, I guess not. All right. So everybody knows English?
Yes.
They understand English completely?
Yes.
Good, very good. So I want you to focus first of all behind Rubina. So that is Rubina, that is Asha. They are also visiting with us, right? She planned the whole thing. And I forgot, Vinayak. Yes, Vinayak is also visiting us. So I want you to focus behind what is written behind Rubina. Why? Okay. I have a 3-year-old kid. My son is 3-year-old, and he continuously asks why. Everything I tell him, why, and that is normally how we are born. When we are born, we are curious, we ask questions all the time. He has non-stop, he gets me very tired by asking for everything. He asks why. But when we grow up, we forget to ask why. Okay? And the only thing I learned when I did all the higher studies, masters, PhD, postdoc, don't worry about all those names. The only thing I learned was to ask, actually relearned, was to ask why. Okay? So if you do not take anything from this talk, whatever I am showing you, just remember that continuously ask why. Fine? Never forget to ask why. Fine? So anything somebody tells you, you should always ask why. Until unless you are completely satisfied, you should not believe it. Fine? Okay. Now is going to be really challenging for me, because I have already told you the secret of finding out the truth. So my title today and the field that I am going to introduce to you is called Nano. Yes, so world is the world we live in, and everything that we are going to see today is going to be nano. Well, why is a good question. So I will start with first of all showing you what nano is, and then we'll tell you why nano is important. So the whole talk, hopefully you will understand why nano is important. Fine? So what is nano? Anybody knows?
Smaller than micrometer. Minute.
Good. Minute is good, but minute is not something which is measurable. What is minute is a relative term. So how can you tell me what's a nanometer like? Okay, how much is a meter? No, tell me in terms of what is that 1 meter long?
No.
How much will be 1 meter long? Is this 1 meter long? Something like that. You see his hand? Something like that is 1 meter long, right? Okay. So what is nanometer then?
It is used to measure. It's smaller than micrometer. It is a study of smaller than.
Is smaller than micrometer. What is micrometer?
It's the to measure the micrometers.
Okay, let me show you. You understand what this means, right? 10 to the power minus 9. If it is 10 to the power minus 1, what does that mean? No, 10 to the power minus 1 means 0.1. Do you know what is 0.1? Right? If it is 10 to the power minus 2, 0.2. Sorry, my bad. 0.02. 0.01. Okay? Shall I write it down somewhere? Actually, you can write it. Yeah. So 10 to the power minus 9 means there are 8 zeros before the 1 after the decimal point. Okay. Now you might be wondering, that's very small, I don't understand what that is. So let me give you an example. Let's say India was 1 meter long, the distance he showed. He showed this much distance, right? 1 meter long. This is India. Tell me how much will be 1 nanometer?
This much.
He is actually quite close. It is the size of a, no. This is India's size. Let's say you take India and you make it this small.
Bacteria size,
Huh?
virus size of a virus. Bacteria size.
Micro.
Forget about virus. Have you seen virus? You haven't seen virus, right?
Microscope.
If I make India 1 meter long, what will be the size that is most equivalent to a nanometer?
If India is 100 nanometers, then 1 nanometer is the size. If we consider 1 meter long and divide it by 100, then we'll get 1 nanometer.
No. Okay, let me try again. He showed you 1 meter. Fine. Imagine that India was only 1 meter long. What will be 1 nanometer long? Here is close but not close enough.
1% of the nanometer.
So the size we are talking about is, if I want to compare India as 1 meter and the size we are looking at is the size of the rice grain. Okay. So if India was 1 meter, the rice grain is 1 nanometer. Do you get a perspective of how different the sizes are? Okay. That's what I was trying to tell you. So it's really, really small. Okay. Now you might be saying, why are we interested in nanometer? Yes. So this I already told you. This is what I am going to show you today. Now you might have of nano technology, you might have heard of nano science. All those things are happening in the world because lot of things actually happen at the nano level. Today I am going to tell you about a new kind of nano world that already has been there, but I will show you ways we can actually now see it. Okay. So this is the length scale that we are going to be looking at. Here is 1 millimeter. Rice grain is about 2 millimeter in size. A human hair, which is about 100 micrometer thick. Okay. So this is plotted also in something called the log scale. Does anybody know log scale? They are not linear. They are, but don't worry about it. You just follow these numbers. Okay. This is 100 micrometer thick. Here is a cell that is inside your body. You do not see it usually unless you put it on a microscope.
It's an animal cell.
Yes, animal cell. You are also an animal, so that's your cell. You have bacteria, which also you do not see unless you put it under a microscope. I will show you some movies of that. These are blood cells, red blood cells, right? This is chromosome. If you go pull it, you get to DNA, which is 2 nanometer thick. Okay. Now we are talking about nanometers. Viruses that you have never seen, but you know.
We have
Inject
seen.
in the people's
It's a
body.
bacteriophage.
That is a bacteriophage, yes. Okay, all these are in nanometers. From this point onwards, everything is in nanometers. These are hundreds of nanometers, these are tens of nanometers, these are a few nanometers. Okay, and you are all made up of all this. But you have believed everybody, you have never seen anything. Everybody told you you are made up of nanometers things, you're made up of protein, but you cannot see nanometer things easily. Have you?
How can we see it?
Huh?
You can see it by
That's what I will tell you today. Okay?
Why will you see it?
Why do you want to see it? So that's something that I will tell you too. In fact, that is the fundamental thing that I want you to take away from this. Why do you care? You are fine, all your hands move in millimeters and you walk in kilometers and your trains and things go hundreds of kilometers, but why should you worry about the nano world?
Because we are giving out of time.
Without knowing fully.
Well, one thing, yes, but it is also much more fun. Okay? Let's come to that. There is other nano world also. That one works inside my computer. That one works on many other things like small gears that are nowadays can be made in nanometer scale. This is transistors that work in my computer. They are actually now being made in nanometer scale. Okay? So, if I keep zooming in and in and in, the minimal thing that I will see is of the order of 20 transistors, which will be of the order of 20 nanometers now. Okay? There are other things, molecules, quantum dots, don't worry about them. These are also part of nanotechnology, but we will focus today on this nano world. This nano world is man-made, this nano world natural. It always has been there. Okay? So, you are nano objects actually being run by nano objects. Okay? So, you are the best kind of nano world. So, what is the difference then between the nanotechnology as people talk about nowadays, transistors, nanoparticles, and what I am going to be talking about, what exists in your bodies all the time? There is one major difference. Do you know what that major difference is? This is true. Actually, machines nowadays use energy. They can produce more stuff, new stuff can be made, and they can do computations. They do all the same things. So, what is the difference? Well, there are very subtle differences, of course, but one difference that is very obvious to anybody, well, you can make new machines from older machines by putting things together. So, they are not growing the sense we divide ourselves, but that is also a critical difference. I will come to that. The biggest difference that you can clearly see is if you just take the nano objects, in fact, you are made up of nano objects, take any grasshopper, take any bacteria, put it inside the freezer, okay, cover it in a plastic bag so that moisture does not, put it inside the freezer, it will stop working. Okay. However, if you take my laptop, you cover it in a bag, you close it, you put it in the freezer, take it out 2 months later, it will work just fine. Yes, it will.
Because technology development.
Yeah, so what is that difference?
No, that is living, this is not living.
Well, yeah, I understand, but somebody told you this is living and that is non living, but you have to understand what is the actual difference between them, and that's what I'll tell you. So, hopefully, I have something new to tell. Yes.
We think life is working.
We think. Yeah, but bacteria doesn't think directly, right?
It is.
What is life, right? So, I will not try to ponder on the question of what is life. Let me try to explain you in slightly different way today. What is different between the two is that one is dependent on temperature, the other one is not dependent on temperature. Okay? So, for example, you are very severely affected if something becomes cold. You don't really want to do anything anymore if it becomes very, really cold. Computer has no problems there. So, this temperature thing is a crucial difference. Okay? Now, yes.
What if the virus attacks?
Yes, but it will not change the temperature directly. If your virus attacks you also, then you will also be sick even if the temperature is fine. So, we are not looking into special cases of that, but normally temperature makes a big difference between our nano world and technological nano world. Example is just as I said, if I cool down, yes, hold on, okay, let me answer her. If I cool down something, then things will stop working. However, if I cool down my computer to 10 degrees, 4 degrees, it will still work. Okay, it will work in the Alaska station, but my body, if I leave it out there, I will die. So from here you can understand there is a subtle difference between our nano world and the computer's nano world, right? This temperature difference is actually also very important in the sense how these molecules inside our body move, like the nano world inside our body moves. The difference is that temperature basically makes everything roaming around randomly, and that's I will show you in a little while. Hence, if you increase or decrease the temperature, this movement of small molecules that are inside your body becomes larger or much smaller, and things which would have worked normally would not work anymore. Okay. So, now let me try to show you this from yes.
We are also made up of molecules and atoms. Even that is made up of
Yes,
molecules.
exactly. Good question.
If we feel cold, our molecules and atoms contract.
Yes.
Even then it contracts.
No, molecules do not contract. What contracts is the distance between them.
Yeah.
So, let me come to that. I'm trying to explain that part to you, in fact. So, I have not solved this problem, but focus on. So, when I said temperature, I also mean heat, right? Temperature means higher the temperature, more heat is there. So, heat or temperature is some kind of energy. You have been told this, and you have also been told maybe that energy is conserved. Have you heard this concept?
Yes.
Okay. So, now let me give you an example of this guy holding a rock and dropping it. Of course, I could have done this experiment, but I couldn't find a rock, right. So, I drop my rock. What does it have originally?
Gravity.
So, U here represents potential energy. Okay. Gravity is the attraction that is pulling this down. So, this energy, when it is dropping down, gets converted at this middle point. It is equal and equal. Half of the potential energy has been changed to half of kinetic energy, and then at this point, right before it hits, its velocity is highest, but the distance is the lowest. It is completely converted into kinetic energy. Doop! It fell. Well, it fell because of gravity, but now it is not moving, so it has no kinetic energy, and it is also not at a height. There is no potential energy. What happened to the energy? Converted to what? Well, it is not moving, so there is no kinetic energy. Yes, but where is the energy? It didn't disappear. She said conserved, but where is it?
Inside the rock.
Yes. Okay. If you touch the rock, you will see heat. It probably not be perceptible to you so much, but if it falls a long distance like a meteor, you see heat. Yes.
Will even have weight also not?
It will not have weight.
Rock.
Rock will have its own weight, but weight is not energy.
I know that. I was saying.
Come again?
But the weight
I
will
was,
be
I was saying that it will have weight also due to which the gravity is the
Yes.
gravitational
So,
force
the
will
reason
increase.
it fell was because it had some weight. When you increase it to a height, it had some potential energy. That energy, if you left it, gravity pulls it down and it gets converted into a kinetic energy. This, when it falls onto the ground, has been converted into heat energy. Okay. Now, everything is good. I understand that energy is conserved. What I do not understand is, if energy is conserved, everything is fine, why does not this rock go back? Well, energy is conserved, so whether I put it here or I put it here or I put it there, I should be able to do it.
There is a force which is existing, which keeps the
Opposite
stone up.
force.
Opposite and equal
Equal
force.
and opposite.
Yes, if there is an equal force, there should always be a chance that it can go back.
It has only
But it's
force
not
to
there.
bring the
But
rock down.
Due to the gravity
The
coming down
gravitation
on a,
force is
pulls down.
keeping
Towards
it
the
down.
earth is smooth.
Earth is smooth. Okay. Now, so first of all, you are right that it keeps it down. Okay. Have you ever been able to use this heat energy to put it back up there? No. So, the idea I am trying to tell you is heat, while this thing did heat up, I cannot use this heat to move it back up. You have hardly ever seen a rock go up by using its own heat. Okay. So, the idea from here is, so friction has converted mechanical energy into a thermal form, and that thermal form cannot be converted back. Okay. So, what happened was, while a total energy is conserved, we have lost something. What is that? No, we did not lose energy. I told you energy is already there. No, heat, well, heat will eventually, if it was in a nice vacuum chamber, then heat will not radiate out. What we call it is the useful form of energy. So, we call it by the quality of energy. The quality of energy in petrol, for example, is high quality of energy, because we burn it and gives us huge amount of energy. Okay. Water, it falls, gravitational energy pulls it down, sorry, gravitational force pulls it down, and you get huge amounts of hydroelectric energy. Thermal energy that you saw in this rock is something that is poor quality. It will not give me anything useful. Okay. And there is a way we describe this also. So, the reason we cannot use it is because this heat energy is in random. Do you know what is random? No.
It varies.
Jumbled necessarily.
Jumbled up and random.
So, these words are close to it. It is happening in all direction at all times. It is not constant in one particular direction. It doesn't even have to be constant in a particular time. Okay? It's all kinds of orientations are being taken by the molecules. Okay? And of course, we have not seen molecules. Have you seen molecules till now? No. So, I told you the rock is made up of molecules, somebody told you or your body is made up of molecules, but you have not seen any molecules till now. You did not, you just went ahead and believed everybody, right? Yes, we are coming to that. Okay. So, hotter the body, faster the motion. They are random, but they are faster. Yes.
Now, why are you talking about energy? Why, how it is related to nanometer?
Let me come to that, because what I am trying to tell you is what is so amazing about Nano-World. Okay? And maybe in 2 more slides you will appreciate why it is so amazing. So, one more thing, this random motion is actually the key to the, but the randomness of this thing, the fact that everything is not the rock when it was falling down, everything was moving in the same direction, right? But now that everything is moving randomly around, it is not usable anymore. So, that is the key for the low quality of the energy. One more thing. Have you heard of this concept?
Yeah,
Free energy. Free energy is useful energy of the system. That is the most easiest way to explain free energy. What does it mean? Remember, this rock has very little free energy, right? When it was up there, it has a huge amount of free energy. Okay? So, that is the energy I have that I can use to do something useful, anything converted to, for example, to drive my car, the batteries charged in my, the charged energy stored in my batteries can be used to drive your, turn on a torch. The way we describe this is, we said this is my total energy and this is my random part of the energy. Okay, I subtract that, and that is my free energy that I can use. Okay, is this clear? Okay. Mathematically, people like to write this equation formula. Okay, this is free energy, this is the total energy, there is T and S. What is T? Nope. Temperature. Remember, it was dependent on the temperature. Okay. What is S? Nope. This is a new term that you probably wouldn't have heard about. It's called entropy. Entropy. Is it written here?
Yeah, it's there. Disordered.
Yes, entropy. Okay. Entropy is this measure of randomness. So, if I have a ball of rice in my hand that is very ordered, that is close together, I throw it on the floor, disordered. Okay. Disorder has, we actually say it in this way, it was more ordered, it is now much more disordered. Okay. Now, there is a key thing that I want you to understand from this equation, that is, if I have a system in any state with any free energy, it will always try to minimize its free energy. I am not going to tell you why on this one, because that needs a lot of more physics, but you have to believe that this is the reason. For example, I take a drop of ink, I put it in a bucket of water and spreads. The energy, I did not change energy, the total energy was the same. So, without changing this, I still got something happening. Why did it happen? Because this disorderedness was increasing. The drop was originally at one point, now it is spreading out everywhere. More spread out is much more disordered. So, I can change the free energy not only by changing the actual energy of the system, but also by changing the temperature or by changing the disorder. At a fixed temperature, I can just change the disorder. And because this is a natural way of going about, everything tends to do that. Everything in the world will follow this rule. So, if I do not change the temperature, if I do not change the energy, it will still disorder. Now, think about all the molecules in your body. All the molecules, you do not add any energy to it, you are at a constant room temperature, they should fly away. Boom, 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 all my molecules in my body should fly away. In fact, do you know how fast the molecules in the air are moving around? You have any idea? Speed, what is the speed? Huh? How many meters per second? Yes. Yes, but what is the speed of this air molecule that is moving right now, which you cannot see, but it is moving? How do you know? That's light speed. In this room, air molecules are moving around at 500 meter per second. That is the speed. I can show you the calculations, but that will be too much for today. All right. I want you to figure out later on, if you have internet access or something, find out how to calculate the speed of an air molecule inside a room.
No, if you type, no, they'll not give the
At a
proper
particular,
answer.
huh?
If you type that, they'll not give the proper answer.
Okay, you can come and visit me. I will explain it to you. Why don't you? Huh? Why should you know? That's a good question. If you increase the? Yes, definitely there I am, remember, I am also adding energy to it. It will move even faster. The total energy has basically increased. But even if I don't add any energy, at a fixed temperature, I will still see random motion.
And now when the room is at fire, what temperature? How fast?
They will move even faster. Okay. This is the reason why, for example, you turn on
All
the
the molecules once
So you turn on a fire somewhere and everything basically explodes if it's in a room, right? Okay. So what I told you was things are moving around really, really fast. Air molecules are moving at the speed of a jumbo jet really. The reason you do not feel them is because when they hit you, they are very, very tiny. So you do not feel their impact on your body. Otherwise, they are actually the reason you have pressure. You fill in gas in a balloon, the reason it holds its shape is because continuously things are striking it. So you can't see the individual strikes of the molecules, but this thing is enough to hold this in shape. You heat the balloon, it becomes larger. That's because random motion has increased. Okay. The reason I wanted to tell you this story was because this is the reason I believe the nano world is amazing. If everything was randomly falling apart, how did we come to be? How do we have the same fingers? Why don't I lose the molecules in my fingers? Right? In fact, if you think about it, if you have heard about origin of life, Earth was all flat and with nothing on it. Right? There was only heat, energy was there, and there were molecules. Somehow magic happened. Did magic happen? Well, I think there's some kind of magic must have happened because no, Big Bang is much before that. I'm talking about Earth has formed. Okay, Earth has formed. I have no humans, I have no plants, I have no bacteria, and slowly.
Water started to rise up.
Yes, but even how did water form? I am telling you everything should fly away. Right? So, I haven't told you a key part of this story. The key part is how do I go from being completely disordered to being ordered? And the way we can do this is by adding more energy. Okay? This is the reason why you eat. This is the reason why you take glucose. Okay? This is the reason why the plants absorb energy from the sun. If they didn't do that, they will start falling apart. So, by adding energy to the system, now I can increase my okay, and minimize also my entropy. Basically, the entropy can be reduced. Okay? So, this is continuously going on in your body as well, and all molecules, even though governed by this universal law of randomness rules, are trying to fight against it to keep you as you are. Till now, I have not shown you any single molecules. Okay. So, the question now we are going to ask is, yes, and we know it is order. So, I will show you at least some pictures. This is a picture of a, it's not really a neuron, it's actually a fibroblast. A fibroblast is the cell that you get from your skin, and it has been colored in many different colors. Okay, the yellow. This is a cell from somebody's body grown in tissue culture, which means you have grown it in a plate. Okay, and the colors have been added. I will tell you how we add colors, but I just wanted to show you the picture first.
So that
I will tell you how do we add colors.
total color will be white only, another means
It will actually be mostly transparent if I did not add this color.
that means we can't see only.
You won't be able to see. In fact, you take some cells, you cut yourself and put it on a slide, you will not be able to see so much detail. The reason you can see so much detail, so you know what this is? Yes. Do you know what these, huh? They are not nerves. These, okay, so you, yeah, it's okay. These, we call them as the highways of the cell. Okay? These are called microtubules. We'll see some of them later on. Okay? Then there is another color. These are called actin molecules. These are called microtubules. These things are called actin molecules. No, it stands for ACTIN. Okay? No, they are not active, they are actin, A-C-T-I-N, N. Yes, okay. But you can see that they have very nice structure. They are not randomly moving molecules, very nicely ordered, okay. Here is something that somebody mentioned before. This is by the way, the scale I have not shown. This is micrometer in size, 10 micrometer in size. This thing are viruses.
No one can come. They'll come tomorrow only.
These are viruses that you already heard, right? What is the size? Do you know? This from this end to this end.
Smaller than bacteria.
About 10 to 100 nanometers, depending on which virus you look at. Okay. Again, they're close to being single molecules. Actually, each color that you see here is one molecule of a protein. Yet they're very nicely organized. Okay. Again, order is there. And this is a key idea of biology. Everything basically can make itself ordered, or we call it self-assembly. In spite of the fact that nature has been telling it that you should just randomly run away. And that's what's so amazing about this. In spite of its inherent tendency for all of you guys to run around in this playground, you still are here, right? We gave you some motivation. Similarly, I can give energy to these and I can ask them to assemble. Okay.
How is that possible?
So that is a very detailed, long story that I will have to tell in a course, but the key idea is written here. First, you add energy to the system and you do it at the nanoscale. One molecule at a time, you bring things together, you make it more motivated to stay together.
So we should just give energy.
We have to just give energy. You pay something for bringing order to the disorder. Yes.
If you don't give energy, will they disorder?
Yes, absolutely. That's what I told you.
When we die, why will our molecules not disorder?
You give it time, it will. Remember, the body disintegrates.
Because they're
Yes.
covered by
Well, the skin is also a model of molecules, made up of molecules. They will also degrade. So,
If we don't let them degrade, will they disorder themselves?
so degradation is itself disordering. Okay, just takes time for disordering. Because they are being held together by this energy. Once the energy is wasted, then you will also disorder.
After we die, suppose we give energy to the body,
Mm-hmm.
will it disorder?
Yes, you have to give this useful form of the energy. If you do it right, yes, on paper, yes, it is possible. But biology is very complex. We still do not understand it so well that we can give energy, right kind of energy to every form of the molecules that we are talking about, and hence we haven't been able to keep life on for ever. Yes.
Snake,
Yes.
if we'll cut its head, no, no?
Yes.
Like rattlesnakes.
Yes.
It will be alive till like more than 5 to 6
Yes.
minutes.
That's because see you are confusing life and energy processes. For example, my body has a lot of energy. You cut my head, my body will still keep doing some things. Yes. The only reason it stopped doing something was because my heart stopped pumping blood to it. Then, as long as you keep giving energy, somebody comes tomorrow and says, you know what, I have a way to connect my head onto my body will still alive if it has not disintegrated. Okay, we have not become so good that we understand how to rejoin those processes, but at the fundamental level, yes, it is possible.
You heard of Frankenstein?
Yes, that is basically the idea. Okay.
Now, after death, does our muscles respond to something?
Yes, they will. They don't care whether they are alive or dead. They are molecules. Okay. The only thing that makes us live or dead are two major things. One is our brain, which thinks, right? And it is probably even thinking when you are in a coma, Okay, it's probably even thinking, we do not know it, even when you're dead. We just don't know it because we do not have ways to measure it. Okay, yes.
Now,
Yes.
I'll be seeing discovery, I'll get many information.
Yes.
Like the hens, they'll peel their skin out,
Mm-hmm.
and then it will be dead already. They'll just put that salt, lots of salt on that.
Yes.
Shake. The
Yes.
body will be shaking.
That is because there is energy and entropy. Okay, so you should not confuse life. Life is a very complex term in the sense that if you have life, it means certain things. If somebody is brain dead, is he dead or is he alive? It's a complex definition of what, or rather I should say life has a very complex definition. So, I will not venture there, but by molecular standards, by just physics of it, everything has energy and everything will have entropy, and those will have it even if your brain is dead.
So
Yes.
more energy,
Supply.
more disorder.
Nope, if you give it the right form of energy, you can remove the disorder. That's what I am talking about. I can make you from your individual molecules. If I give the right kind of energy to the right kind of molecules, I should be able to recreate you. Okay? It is complex, it is not possible physically by me, in fact it is not possible by any person at this moment. But at small scales, we see this happen, we know we can do it.
Can we supply artificially blood to our body? When
That is happen all the time, you can do that. But blood is not the only thing that you need, there are so many other things, that is why it is complex. Okay? But at the simple level, molecular structure. So now you are asking very complex structures. We have not gone that far. We have not advanced so far. Let's come down to the cell levels, the molecules that you see in here. If you do what I told you at the nanoscale, you should be able to create order. You will be able to create all these structures outside, in your lab, in your classroom if you want. I wish I could bring some things for you, but if you guys want to visit, I can show you. Okay? So, life then is going to, so I shouldn't be saying life, in fact, everything you can convert disorder to order by putting more energy. Life also does exactly the same thing. Nature basically does exactly the same thing to create you, me, viruses, bacteria, everything. And it all has to do it in clever ways. Because if you just provide any kind of energy, it would not work. You have to provide the right form of energy, then it will be able to convert it.
what is that energy?
That is energy of highest quality that we talked about before, right? If you just provide heat energy, it will not do anything useful. And it is also should be usable by the molecule that we are talking about. Okay. And we are basically trying to answer all the kinds of questions you asked. And we do this by looking at the nanoscale. More importantly, I want to show you that we can do this by looking at the molecules I was talking about. Okay? That's what my lab does. But this is what keeps me motivated all the time, because this amazing world should be falling apart, but something is coming in, you add some energy to it and it's making something new. It's happening at very small scales for my purposes. It doesn't explain how all the big questions that you asked works. Some other scientists are working on the bigger questions as well, and everything is being tied together into a larger idea of how life works, but we look from this angle. So, how do you see the invisible molecule? Yes, of course. You light them up. If you are in a dark room, if you want to see something, you throw light on it.
So we add
But other way of doing this is I attach a bulb to you and you switch on the bulb, and wherever you move around, you will carry the bulb with you and I will be able to see you. Yeah, so the way analogy I am trying to draw is, if you put a light on the molecule, you should be able to see the light even if you cannot see you.
So we just add colors to the molecules.
Yes, so what we see is the light coming from the molecule that I have attached. In fact, it is usually a molecule itself that we have attached to the molecule of my interest. The molecule of my interest was invisible, but because I have put a light which is visible to me, I just follow the light around and I make sure that the light is attached to the molecule, that somehow very convincingly attached to the molecule I want to study. Okay? That is the key idea here. And if I believe that when I am following the light, I am following the molecule, then we are looking at the molecule. And the trick to do that came from this. Anybody knows what is this? This jellyfish. So, you see this light which is on the rim, yes. That is coming from two sets of proteins. One of them is generating energy, the other one is generating light. Okay. And what great scientists did was they took the molecules from the jellyfish and they figured out ways that they can produce it in anything. They can put it in mice. You see these eyes? This is a regular mice. These mice are fluorescent. Their tails are also fluorescent. This protein is called the green fluorescent protein. It got a Nobel Prize 5 years ago. Okay. Yes.
Will the mouse be alive or not?
It is alive. This is alive. I can put GFP in your body. I am not supposed to, but if I put it on your body, you will be fluorescent. You go to a dark room, you will see. Hey, you have seen some similar phenomena. There is a firefly. It doesn't use the same molecule, but it is fluorescing by. We called it a slightly different name, but it is the same kind of a phenomena.
It does not infect us.
Huh?
It does not infect us.
No, this will not infect you.
Lifespan won't change.
We make sure that the lifespan doesn't change. Of course, that's why we do not do it in humans. Huh? We do not do it in humans to make sure that there are side effects. There might be some things which we do not know, so that's why we do it always in the mice. But it is a great tool, for example, in cells and bacteria, to study and understand how molecules are doing the things I told you, how they are putting together order. Okay? So, these are cells that you saw before. This is bacteria. So, each of these rods that you see are bacteria. Okay? They are what?
They are disordered.
They are also disordered, yes. And we are not going to go into their disorderedness because that's a different story altogether. But good that you are noticing these things. They will be. In fact, these cells will also be disordered technically. They do a lot of work to bring together themselves. Okay? So, good. I'm glad you guys are getting the idea. In fact, you can get, yes, sorry.
Eels.
Huh?
Electric eels.
Yes?
Electric eels
Yes.
can kill there's some crocodile also now.
Yes.
They showed in the video. Okay, what is
No, it's, yeah, what is the question? Yes.
Because like it wanted to eat that electric eel,
Yes.
it had put in the mouth. So the crocodile,
Yes.
it full like balloon.
Yes.
It was floating on the water due to that eel.
I didn't understand the question. What is the question? Okay.
Extra information.
Okay.
Lots of electricity can kill
Yes, of course.
dangerous animals.
See, we are not talking about killing, as I said. The life can be killed in many different ways. But at the molecular level, that is not dead. Everything is still behaving the same laws of physics.
Let's not divert
Okay. Fine. How much time do I have? Okay. It says 46 minutes on my clock. But the good thing is, the thing that I did here, I could also do on the single molecule, just way I described you. I am attaching my glowing molecule to my molecule of interest, and I can now see the molecule that I was interested in.
Why can't we do it on
It's basically, as she was saying, is there any side effects?
Take Himalayan then.
Yeah, so I don't know yet. All right, we want to be very careful when we deal with humans. I don't want to run into a situation even 20 years from now where I made a change in you, and that change your life of that life you are going to have. So that's why we do not do in humans, right?
replace it,
Yeah.
replace it again.
Replacing is possible, but these are tedious. So till we come to a situation where we can easily replace it, we want to make sure that we do not do it. and we would rather do everything in these. So, as I said, we are advancing, but we are not advanced so much that we can do all the things that you guys are already saying. Okay, we would love to, right? That's why we need people like you to be doing these things. So, what I was saying was, you can actually not have one color, you can have multiple colors, by the way. So, there is no limitations on how many different things you can color and how many different things you can see. Okay? But the most important thing is, even inside a single bacteria, I can put the torch on one molecule and they will show up as one. So, now the whole bacteria is not on, only a spot inside is on, one bulb is on. That is because there is only one molecule carrying that bulb.
Won't it reproduce?
Which one? The bacteria? Yes, it will. The bulb. The bulb will be made, new bulb will be made, yes. Actually, this bulb attached to the molecule of my interest is inside the bacteria, it is what we call coded inside the bacteria. So, bacteria is making that bulb, the GFP protein, okay, attached to my molecule of interest. And new proteins are being produced, they also are fluorescent. We call them fluorescent, okay. Fine? So, just imagine that every time my cell's factory is making a new protein, it is also attaching a bulb to it. So, all those molecules are fluorescent now, and I can see them. Our cameras are sensitive enough to detect the light coming from this tiny nanometer bulb. Yes?
If the bacteria reproduce, will the reproduced bacteria also have?
Yes, absolutely, because we have actually put this on the chromosome of the bacteria. Yes, it does. Okay. You can do it in different ways. So, the technical details I have skipped for mostly, but yes, you are right. Fine. All right. So, what can you do with single molecule kind of tools like this? Of course, you can follow them around. That's the easiest thing to do. So, you can say, hey, how much time does it take for this molecule to move from this place to that place? And here's a question that we have asked before. We say, no, it looks like a ribosome, but it's not really a ribosome. What is this one is showing is the nuclear membrane, yeah, endoplasmic reticulum and the nuclear membrane. These red dots are proteins that are supposed to bind to the large DNA inside the cell. Okay, and each time it tries to find this, it can find it in many different ways. I have this spaghetti noodle that is inside my nucleus, and on there I have to find a place where I need to do my job. So, the way it does it is sometimes it randomly binds. I told you randomness is a very important thing here. It will randomly bind, or it will randomly bind at some places and run around randomly, up and down, up and down, up and down, till it finds what it is looking for. Sometimes it will hop from here to there to there. We can figure out all those things by just looking at timing, saying how much time does it take for go from this point to this place, and we can measure those as a function of time. Here is a movie to show you that you can really see the single molecules that I was talking about. Remember this randomness? They are randomly moving around. All these bright spots that you see are single molecules, light coming from single molecules rather. Okay? And what we are drawing here is the path one single molecule took to move around from here to there to there. You see all of them are doing all sorts of different kinds of random motions, right? So, now you are watching the Salman Khan of molecule world. Okay. You can do this. This is a mammalian cell. The inside the nucleus of a mammalian cell, the cell is not visible. Okay. Here's something similar happening inside bacteria. So, these are the two bacteria, and you can see the molecules basically move around in this bacteria. All right. It looks, it's colored, so don't worry about the color yet, but I can put any color I want because I decide which color of protein I add to it. Okay. So, this is just a representation. The way we collect data is only in terms of, it's a black and white movie, but we color it any way you like. Okay. In fact, we are so good at it that we can watch a protein which moves step by step in one direction also, instead of doing random motion. So, here is a molecule which moves like this on the highway tracks that I told you about. I can measure the time dependent changes in distance. It is taking one step. So, this is distance. Look at the scale, nanometers. It goes from 40 nanometers to 80 nanometers. So, it is taking about 40 nanometer steps. 40 to, okay, so maybe 70, something like that, right? Yeah, within some. So, we can measure each single molecule's steps. We can count. Now that we have gone down to the single molecule level, there is no ambiguity of how much. I know I can count here that many number of molecules are there. Okay. So, how do we do that? So, I told you I have given it one bulb, right. So, one molecule carries one bulb, and if I ensure that, then I just watch how many times it takes me for this bulb to go off. In fact, if it is one molecule, this red one is one molecule, it will go off, it becomes 0 and it will show nothing else, because I can't, unlike the bulb that you can carry where you can give little current and reduce the intensity of the bulb, here the bulb is on or off. We call it digital behavior. If I have two molecules, see this line, 2 steps. I do not know when it photo bleaches, we call it photo bleaching, when it dies, but I know that they are going to show 2 steps. There is a molecule which did 4 steps. So, I know there were 4 molecules there. So, I can, even though I cannot see those molecules directly, by measuring the light that is coming from them, I can say how many molecules there were. I could track them also, right. So, people use this to find out, for example, if it is a complex structure that I have created by ordering something, how many molecules there are, how is it changing as a function of time. As we were saying, assembly, how was the assembly taking place as a function of time. We can do even better. We can measure very small distances on the same single molecule. Okay? Come again? The trick we use is it depends on another physical phenomena which happens if I put, instead of putting one bulb, if I put 2 bulbs of different colors, there is a physical phenomena that happens, that if I bring one bulb close, then it starts, even though I am turning this bulb on, this bulb gets energy. Okay? So, that I will have to explain a lot of detail. So, I am going to skip that part yet, but this phenomena basically happens even when, see, the energy is totally conserved, fine, and you can find many ways of transferring energy. So, what is happening is, I am taking the energy that I supplied to this one and transferring some energy to this one, okay, without even touching each other. So, as this comes together, more and more light starts to show up on this molecule. So, I can use this distance based difference in energies to tell how close or how far they are. So, if they are very close, all the energy that I provide to the green bulb will show up in the red bulb. If they are very far, all the energy will show up in the green bulb and total energy is conserved. So, very little energy will show up in the red bulb. I measure the light coming from both bulbs and I should be able to tell whether they are close or far. Okay? The plot that is shown here, the graph that is shown, shows that this works between 0 nanometers to 100 nanometers, perfect for my measurements.
The red dot will accept all the energy from the green dot.
Yes.
The green dot will
not emit any light, because light is also a form of energy, right?
Will it take back the light from
Not while they are close together. It is happening, actually I should correct myself, it is also happening, but as quickly as it goes to the green, it also comes back to the red. So, what we call is the equilibrium has been reached, where the red has most of the light and the green has very little of the light. Okay. And you can do experiments using this. Here is an experiment that I did long time ago, where I have a small strand of DNA. This is not a double helix, this is a single strand of DNA. And there, if I put this protein binds to it. And if it binds to it, it either gives it this kind of configuration or this kind of a configuration. You can see the distance between the two bulbs have changed, right? So, I should be able to see this. And if I just look at the light coming from one molecule like this, what do I see? The red bulb goes down in intensity, the green bulb goes up in intensity. Why does it do that? Remember, energy is conserved. So, if I add the two energies together, that will be constant. Okay. So, donor is basically the green bulb and the acceptor is the red bulb. Fret is the phenomena that we are talking about. So, I am keeping out all the technical details, okay, because you will probably see them only in college or later half of your college. But I wanted to still show you the data. This is raw data in the sense that this is the actual experiment. Okay. Okay. This is really the light I have measured from one bulb, tiny nanoscale bulb. Okay. And it changes because this kind of switching between the two is happening. And then I can calculate the distance based on this kind of from this I can do that. So, it is going from being really close, far, close, far, close, far, as it switches between the two as one protein comes in and binds to it. Okay. So, we can not only see the molecules, we can measure distance changes within the molecules. Yes.
What will happen if there would be another
protein comes in? In this, it cannot bind because there is no more space for it. But when this falls off, that can take over. I wouldn't know the difference. For me, I am only measuring. So, it's by designing the experiments right, we can understand what we are interested in. That is also true. The other thing we can do is even better than actually seeing the molecule. We can now play with the molecules. So, I can pull and push and press single molecules.
No, but it's very
tiny. I agree. Yes, in fact, it is a problem because thermal motion tells you it will be randomly moving around, and the energies that will be required for holding them will be very, very small, right? But we have come across situations like this before. Okay. We can actually now attach one end of the molecule to a surface and the other end of the molecule to a larger molecule like a bead, which is micrometer in size, and we just observe the position of the bead. Okay. So, here is how we do the experiment. We attach, so this is a complex molecule. Here the same kind of a protein that you saw before is bound to the small single stranded DNA, and then one end of the DNA I have attached to the surface, the other end I have attached to, so these are not to scale. This bead is much, much larger than this, about 1000 times larger, 10,000 times larger than this. This is in micrometers, this is in some nanometers. Okay. If I take this bead and I shine laser on it, a phenomena that I am also not going to explain to you today called laser trapping happens. Have you seen Star Trek?
Yes.
Star Trek has this beam shown at the this starship, and then it catches the starship and pulls it. Tractor beam. Yes. The idea is similar to that. We do the tractor beam on one 10 micrometer object or 1 micrometer object. Okay. If I now move this beam or I move this stage, then I will be pulling like this. So, I can apply force on it and see how things move, and you might be wondering why would you do that. So, I will come to that later on. But here's an example of how it will change a molecule like this. As I am opening, these two things will go far away. Do you agree? Yes? So that's like normal machines. So here the gray thing is how force is changing. I am increasing the force. What happened?
She wanted to go to washroom.
Yeah, go ahead. Yeah. Anybody else wants to take a break? Sorry. Yeah, go. I'll show you what we can do if we make mutations like that. Here is a tool that I showed you where I apply force, and as I apply force, this is the signal which tells me the distance between the two. The distance between the two is increasing. So it is inversely proportional, means as this thing goes down, then this is basically going from this to being this. And you can keep repeating this. This is happening on one single molecule. I am doing this. All right. And this is the raw data from which it has been calculated. Now, what can I do with it? So here is a situation where we have taken the two bulbs. The bulbs are now shown in yellow and blue, and they have attached them to a larger molecule which connects the cells to the surface. You saw the cells were attached to the surface, they were spread out. These are the molecules which actually keep them attached to the surface. And when I put this mutated protein inside the cell, I see color from this, I see a color from this. And I can measure how that color is going to change between the two as distance between the two. So if this thing stretches out, the more color will be visible from this one. If it is closed down, more color will be visible from this one. That's how we are going to see this. And if you put them inside the cell, you can see the cell here. Now you do not see the whole cell, you just see the molecules which are lying at the periphery of the cell. You see these, so this is not so clear, but it zoomed in images, you can see different colors. These colors are for different amounts of distances between the this molecule to this molecule, different amounts of stretching. So somewhere it is less stretched, then somewhere in the middle it is much more stretched. So this is the bar, here is very little stretched. Okay, so inside the cell I am measuring the force the cell is feeling. Not your body, a cell's body, how much force is it feeling. Much more fun would be actually to show you the movie. So here is the cell, there is one more cell here, see this, these are the legs that it spreads out, this is another cell, okay, and watch the movie. Okay, as it's retracting its thing, the forces here are changing. So we can measure them without actually touching the cell. We are not touching the cell anymore. This is not an amoeba, this is actually a cell which is similar to your skin cell.
Is that the microtubule you're looking at?
No, we are not looking at the microtubule. This is a vinculin, yes. These are vinculin molecules which have been fused between these two.
If it is red and orange,
Yes.
and if they come too close, won't they mix up and become
Some different color.
some different?
Yes, you are right in that sense, but remember this is a pseudo colored image in the sense that I am scaling the distance between them in terms of color.
It's disordered.
Okay. Well, it is not disordered so much anymore. Okay, they are changing their things in very well defined forms, and how it does it is what we are interested in spite of being able to be completely disordered. Okay. We can do something else called super resolution optical microscopy, and don't go by the name. This is the one which got Nobel Prize this year. Okay. The idea is, I showed you images of single molecules even in the bacteria, right, and you saw a big spot, large spot. The bacteria was 1 micrometer in size, the molecule I told you was of the order of 1 or 2 nanometers in size, but you saw the spot which was almost the size of the bacteria. And this is a phenomena called optical diffraction limit. You cannot resolve much better than 200, 300 nanometers with regular light of any color that you normally are used to seeing. Okay? Whenever you take a molecule, if it is, even if it is lying in this center, right, 1 nanometer, it will still look to you like this. The pictures that I showed you on the camera will look something like this, even though the molecule is sitting right in the middle of that. So the bulb might be very small, but the light will glow in every direction. Okay? So that was a major problem for a long period of time. All light based microscopy that was being done was limited by then. Now you can of course change, as I said, it was dependent on the wavelength of light. People said, you know what, just change the wavelength of light. So if you go to electron microscopy, where the wavelength of light is much smaller, you can now get very high resolution images. The only problem with electron microscopy is you have to do very special processing. You have to fix the cells, you have to kill, you have to add something to it to make a contrast and then take images. So it doesn't show me all this nice moving dynamic movies anymore. So we wanted to make it so better that we could do it using light microscopy or fluorescence. The trick they came up with, they said, hey, we can already see single molecules, and I know how the single molecule is supposed to look like because I understand the emission of the light from a single molecule very well. It's one molecule. The light emission would look like a mountain to us if I look at how the light is spread around the molecule. And then all I need to do is find where the center of the mountain is. And you can tell the center of the mountain very nicely here, the peak. So they take now images of single molecules, they just measure the peak. There's one more thing that is a problem here. I told you there are many hundreds of molecules inside the cell. The only reason we saw one single molecule inside that nucleus moving around was I had artificially colored only a few of the molecules that were there. This will not be the case if I want to take an image of the whole cell. There are so many molecules. So the second trick they did was they said, let me switch on only one bulb out of 1 million. So 1 million population, one bulb switches on. Now it is going to be only one molecule within the city. Then I say find its center, much better than what the actual image is. Then I switch off that bulb, turn on another light, switch out its center, and I continuously keep doing this very fast. So that's basically shown here. I have control now. I am providing another laser, not the original light that I was required. Another light, when I switch that on, the molecule turns on. Switch it off, it goes away. I can continuously do this. So if I do that, I have now control over when the light is getting turned on also. So this is the idea. You have a molecule, if all the light on this is switched on, it will be one large red spot here. But if I switch on one light, then it will be like this, and I can find its peak. That's right here in the middle. You can find the middle of that. Switch that light off, and then turn on another light, find its peak. And you continue to do this till you recreate your image. This is much better than if all the lights were turned on, which would have been all red here. This is the concept of this year's Nobel Prize. Okay. How does it work in practice? You will recognize the structure, but things are, so this is of course our Eiffel Tower was not lit up like this, but this is to just show you that we collect lots of movies like this where blinking molecules are turning on and off, on and off. We find their centers, we plot those, and we recreate the image of the sample we have. And using this fluorescence molecule, this is basically how a bacteria would look like normally. And let's say I color a few of these molecules which I am interested in. Let's say in my case it's a RNA polymerase. If I just turn on my regular microscope, I will get an image like this, but with my new microscope that I have made, I will get an image like this. So, now I am not going to go into the details, because it is a lot of statistics that we do, but we can tell you that this is a single molecule of RNA polymerase inside a living mammalian cell. Okay. So, now we can see it, we can measure it, we can track it, we can pull it. What does it tell us? So, I want to show you some examples where this disordered thing is now working hard to create order. Okay. Here is one example. So, this is something that people knew a long time ago. I have muscles, I can continuously keep doing this as long as I have energy. Remember, this is the easiest form of order, right? I am at will changing between the two. It is not because your brain is telling it to tell, of course that controls it, but the reason you can do this is because there are things happening which are moving in ordered fashion rather than disordered fashion. Otherwise, my hand will be going all over the place. So, if I just take this muscle, take some cells from it, I will see these kinds of bundles are there. If I take it even further, open up one bundle, I will see that they are also made up of more bundles. These bundles are called myofibrils, and what we see is some dark and some light patterns. I open that and I see there are some really strong, these dark patterns are made out of some bundle of many molecules of the same kind, and this is thin one is another bundle of some other kind. Remember, I was telling you about, if you want to do something, better do it at the nano scale. So, the reason your hand moves is because something is happening at the nano scale. What happens is, these dark ones can hold on to this. Remember, we were talking about the actin molecule. These is a filament, a polymer of the actin molecules. This is molecule is called the myosin molecule. These heads that you see on them, they actually attach themselves to this, and then it's almost like that, but this case the golf, unlike the golf which hits the ball, this one changes like this. All right. When it changes, in fact it has two heads, so it does this, and all of these do this. Hence, this thing moves with respect to this. So, this one will move inside, this one will move this way, and then when it needs to relax, it will move the other way. You are getting organized motion. Every one of these heads is going to use our energy currency called ATP, adenosine triphosphate. Okay. Now, I do not go to the muscle, I say I already know the molecules, I take the molecules, I throw them. In this case, I have done a slight difference between what the muscle does and what we do. We have attached these molecules directly to the surface of my glass slide, you can think of it in this mat, and then taken these bundle of these and thrown them onto the surface, but I have also colored them. Now, they do not have anything holding them, so what do? They move around, they are just continuously using energy to move around, keep running around. All right. So, we can actually study them, how they move around, how much energy do they need, everything that we need to know, how they polymerize, just by looking at these single molecules. In fact, we can do the other experiment also, where we can color those and watch them. I will show you some similar experiments later on. Here is another molecule that you know of very well. Okay. You know the name? It's okay if you don't know the name. DNA is what forms all of your genetic material, all the code that you carries to making proteins and so on and so forth. This DNA molecule is actually a very flexible molecule. You know why it is a very flexible molecule? A large molecule will not want to be large and long. It will want to be doing this. Why? Because this is much more entropically stable state. Entropy is high here. How many ways can I make a linear strand? Only one way. How many ways I can have this random strand? Many different ways. Yes. Okay, sorry, I really overshoot all the time. So let me show you just the movie. What is happening is new DNA molecules are coming out of a virus. And there is a flow that has been attached to this chamber. Okay, and so the DNA is being pulled. It's not random anymore. It is pulling in this side, and as the condition is changing of the solution, new viruses are ejecting more and more DNA into the solution. So you can watch that. The reason I wanted to show you more importantly was this is a very flexible polymer. If I stop the flow, they will just coil back and become a round ball. Okay?
How are they getting pulled around?
Because I have applied some flow on it. Just like you are swimming in the river and I have attached you to the rod and then flown the river, started the river, your legs will be flying. That's the same thing that is happening on this. Okay, I can not only watch the DNA molecule, I can watch molecules which are moving on the molecule. Okay, so there is so this is the DNA molecule. In another color I can see molecules which are sitting on the molecule. These are protein molecules that actually do this search that you saw before. They were searching for things moving around on the DNA molecule. Here the DNA is long and linear, hence they will move like this. I can plot those and I can study that. Here's another case. These are these motor proteins that I told you about, and actually I told you you can put them in many colors. So there is actually some red colored molecules. The red ones are called kinesin. They move your cargo towards the nucleus, and there are the green ones which are called dianines. They move them in the opposite direction. So you have transporters of both kind. Okay, saw that some of them were going in one direction, the other one is going in. Let me play it one more time. Okay, these are walking on those linear highways that you saw before in the first of those movies. And I can pull each of these motors. By the way, one motor, this is one motor that is moving. I can pull on one motor by attaching a bead, just that same kind of an experiment that you saw. And if I do not put that laser, then it will be randomly moving around. Okay. If I put the laser on, the laser is trapped. Okay. And then I put, you see that line? That is, now the laser is actually holding the bead in its place. So while it tries to go there, because it has energy it's using energy to move, it's applying force, the laser is pulling it back. How much force can it apply? I can measure all the forces, individual forces. These forces come in, not Newton, not micronewton, not nanonewton, but in piconewtons.
What is Q?
It's 10 to the power minus 12. Okay? Okay, I will skip this. Let me show you this. Oops, sorry. Here is a technology that allows you to see as new proteins are being produced inside the cell. Every time a new protein is produced, there will be a flash of light here. Just watch the movie. So these are bacteria, they are dividing, and once in a while you see some flashes of light. Well, yes, binary fission is right, but what we are interested in is these protein flashes that are produced. We can watch every time a new protein is being made inside the cell. Okay. Okay, I'm going to skip this one. Is that all right, or what? We are out of time already. This is the molecule that makes ATP inside your body. Okay, It's the energy making machine of your body. It's called adenosine triphosphate synthase. This molecule is called adenosine triphosphate synthase, and it rotates around generating ATP in 3 steps. So it moves like this. The way they watch it is they attach a long rod to it, the same kind of filaments that you saw before, and they just watch the rod rotating around. Those pictures are here. You can watch it rotate. Rather than walk forward or backward, it keeps rotating. It's your own ferry go around. All right, I'm going to skip all this. I will just tell you about the last thing. There is another reason you want to do these kinds of measurements. If you just measure the total number of protein of a particular kind inside bacteria, yeast, the cells that you mostly study, if you just measure their numbers, you will find that they are actually in very low numbers. So this is for bacteria, the most of the time, this is what we call a histogram plot, where this shows how many proteins have 10 copies of the protein inside the cell. Most of them have only 10 copies. Only 10 proteins of a certain kind inside one bacteria. 30% of them have less than 10 copies. In fact, many of them have only 1 or less. What do you mean by less? There are many bacteria which do not have that protein. Okay? So, single molecule can actually make a large difference on the behavior of the cell. Okay. I just want to thank the people because I did not put all references there. This is not all my work. It is work done by me in laboratories of many people. There are data that I took from my friends. Okay, and I want to thank this guy who writes a very nice book called Biological Physics, from which I kind of showed you the ideas. Okay. Thank you. But yeah, thank you for listening, and thank you for bearing with the time. I always get carried away.
No, the talk was very nice.
Okay,
Only
good.
It was
Too long.
divided in 2 different talks.
Okay.
It was really nice.